Welcome to the explainer. Right this second, trillions of cells inside your body are dividing. It's happening constantly. It's how you grow, how you heal, how you, well, exist. So today, we're going to zoom way in and take a look at this incredible engine of life, cellular reproduction.
All right, so here's the plan for our journey today. We're going to start with a really basic question. Why do cells even bother dividing? From there, we'll get into the mind-blowing problem of how they pack up their genetic blueprint. Then, we'll walk through the cell's entire life cycle step by step. And finally, we'll check out the amazing quality control systems that keep everything in check. And yeah, what happens when it all goes wrong.
Okay, first things first. We have to tackle this huge problem that every single living thing has to solve. And what's wild is that it all comes down to some pretty simple physics and geometry, but the consequences, they're absolutely massive for biology.
So, have you ever wondered about this? Why are we made of trillions and trillions of tiny little cells instead of, just a few giant ones? I mean, wouldn't that be way simpler? Well, the answer to that question gets right to the core of why cells have to divide in the first place, instead of just growing bigger and bigger indefinitely. And the answer, the whole secret, comes down to this really important ratio. Just imagine a cell's surface, its membrane, is like all the doors and loading docks of a factory. That's how all the good stuff, the nutrients, get in and all the bad stuff, the waste, gets out. The volume, on the other hand, is the factory floor itself. And here's the catch. As that factory gets bigger, the amount of factory floor space grows way faster than the number of doors. And this image just shows it perfectly. Look at that small cube. It has a great surface area to volume ratio. But as it gets bigger, whoa, look at that ratio plummet. The volume just explodes, but the surface area can't keep pace. It really is like trying to run a massive warehouse with just one tiny little door. It's a logistical nightmare. It becomes super inefficient, and pretty soon, the cell would literally starve itself. So what's the solution? To grow, you can't make bigger cells. You have to make more cells.
Okay, so we've established why a cell has to divide. But before it can do that, it runs into another incredible, almost impossible sounding challenge. It has to make a perfect, flawless copy of its entire instruction manual, all of its DNA, and give it to the new cell. And the problem is, that instruction manual is just ridiculously long. And get this is where the scale just becomes insane. If you stretched out the DNA from a single human cell, it would be about 5 centimeters long. That's almost 2 inches. Now think about this. All of that information has to be perfectly, neatly packed into a nucleus that you can't even see without a powerful microscope. How in the world does the cell pull that off without it becoming the worst tangle you've ever seen? Well, it uses this absolutely genius packaging system. So first, the long DNA strand wraps itself around these little proteins called histones. It's kind of like winding thread around a bunch of tiny spools. Then, this whole string of beads thing coils up on itself. And if that wasn't enough, that coil then coils up again, a process called supercoiling. If you've ever seen an old-school telephone cord twist up on itself, you know what I'm talking about. And the end result of all this coiling is the super compact, super organized structure we call a chromosome. All right, so we've got the why cells have to divide, and we've got the what they do with all that DNA. So now, let's get into the how. We're going to look at this repeating, almost dance-like sequence of growth and division that we call the cell cycle. This right here, this is the cell's entire life story in one picture. And you know what's kind of surprising? A cell actually spends most of its life not dividing. It's in this really long phase called interphase. You can think of this as its day job phase. It's just growing, doing its normal cell stuff. This part is broken down into 3 stages. First is G1, where the cell just gets bigger. Then comes the S phase. S is for synthesis, and this is the big one. This is when all of that DNA gets copied. And finally, there's G2, which is like a last-minute check to make sure everything is good to go for the main event. And speaking of the main event, here we are. After all that prep work and interphase, the cell is finally ready for mitosis. This is the big show. It's this incredibly precise, choreographed dance of separating all those duplicated chromosomes to make sure that each of the new cells gets a perfect, identical copy of the genetic blueprint. Now, in reality, mitosis is one smooth, continuous process. But to help us understand what's going on, scientists break it down into 4 distinct stages. Just think of them as acts in a play. We've got prophase, metaphase, anaphase, and telophase. Let's take a look at what happens in each of these acts.
And boom, here's that beautiful dance in action. It kicks off with prophase, where the chromosomes all condense and become visible. Then comes metaphase. See how they all line up perfectly right down the middle at the cell's equator? The real drama happens in anaphase, where the copies get yanked apart to opposite ends of the cell. And it all wraps up in telophase, where two brand new nuclei form around each complete set of chromosomes. It is just a system of breathtaking precision.
But hold on, the job's not quite done yet. After those two new nuclei are formed, we still have one cell. See, mitosis is all about dividing the genetic material. Now, the rest of the cell, the whole thing, has to split in two. And this final step, the splitting of the cytoplasm and all the other goodies inside, is called cytokinesis. And it's pretty cool how this final split happens actually depends on what kind of cell we're talking about. In animal cells, like ours, the membrane is flexible, so it just sort of pinches inward, forming what's called a cleavage furrow. Imagine pulling the string drawn on a bag closed. But plant cells have that tough, rigid cell wall, right? They can't pinch, so instead, they build a new wall right down the middle. This structure, called a cell plate, starts in the center and grows outward until it separates the two new cells. Okay, so you can probably tell this whole process is unbelievably complex, and you just can't let something like this run on autopilot. I mean, think about it. If even one mistake is made, say a chromosome doesn't copy right, the results could be absolutely catastrophic. And that's why the cell has this whole sophisticated system for quality control. So who's the conductor of this whole orchestra? Well, a big part of the timing is controlled by a group of proteins called cyclins. You can think of them as the green light signals. Their levels in the cell go up and down, and when they reach just the right amount, they team up with these enzymes called CDKs. And it's that pairing, the cyclin and the CDK getting together, that's like turning the key in the ignition. It's what pushes the cell forward into the next phase. But, just as important as the go signals are the stop signals, and these come in the form of critical checkpoints. They're basically like quality control stations on a factory assembly line. They are constantly monitoring everything, asking really important questions, like, hey, is any of the DNA damaged? Or did we copy all of it correctly? Or are all those chromosomes lined up where they're supposed to be? And if the answer to any of those questions is nope, the whole process slams on the brakes and waits until the problem is fixed. So what happens if the problem can't be fixed? What if a cell fails a checkpoint and the damage is just too great? Well, the cell can trigger this incredible process called apoptosis, which is basically programmed cell death. It's kind of a noble act, really. This cell recognizes it's faulty and it self-destructs for the greater good of the organism. It takes one for the team. And that brings us to our really critical and often tragic question. What happens when this amazing, intricate system of go signals, checkpoints, and self-destruct buttons breaks? What happens when a cell just completely loses control? Well, that failure of control, that's the very definition of cancer. When you get right down to it, cancer is just uncontrolled cell division. This chart lays it out pretty clearly. Normal cells, they play by the rules. They listen to the signals, they stop dividing when they're supposed to. But cancer cells, they blow right through every single stop sign. They just keep dividing and dividing, piling up on top of each other and pushing out the healthy cells around them. And this image, this shows you exactly what that loss of control looks like. You can see the cancer cells just growing and dividing wildly, with no restraint at all. They form these tumors that literally invade the normal healthy tissue, stealing its resources and messing up its job. So what causes this whole system to break down? At its core, cancer is a genetic disease. It's caused by mutations, which is just another word for damage, to the very genes that are supposed to make those stop and go proteins we were talking about. Now, sometimes this damage can be inherited, but a lot of the time it's caused by stuff in our environment that we call carcinogens, things like the chemicals in tobacco smoke, or even UV radiation from too much sun. Which kind of leaves us with this one last big thought to chew on. This fundamental process, the cell cycle, is what builds us, what heals us, and what allows life itself to continue. It's amazing. But it's also a double-edged sword. Its precision is literally the engine of life. But when it fails, it becomes the root of one of our most devastating diseases. And really, understanding that duality, that balance, is the key to understanding both health and sickness.